So Keynote 427 was a trial of single agent pembrolizumab, a PD-1 inhibitor, and it was given in the first line to patients with metastatic renal clear cell carcinoma. This actually surprised me. It produced a much higher response rate than I expected, and from that perspective, I think it indicates that single agent checkpoint inhibitor is very active in the first line. Now, putting this in context, we have a situation where we have trial data of combinations of a PD-1 and a CTLA-4 with nivolumab and ipilimumab that has just been approved by the FDA in that setting. And we also have data that are evolving and likely to be presented for FDA or EMEA approval in the near future of a combination of bevacizumab and atezolizumab. And from that perspective, the combinations looked like they were going to hold sway. But as we've seen in melanoma, the outcomes from single agent therapy may be less toxic, less expensive, and just as good at the end of the day. So this raises some interesting questions in the first line. And we need to pursue what the activity of single agent checkpoint inhibitors is likely to be with some other trials. To place that in further context, the next wave of data we're going to get compared generally with the TKI sunitinib will look at a combination of these agents with another TKI, typically axitinib. So we're going to have a raft of data, and sometimes we move a little bit too fast, and we need to really go back and see whether what's a pretty well tolerated single agent checkpoint inhibitor therapy can be as good as adding other drugs. So that is actually very thought-provoking. It's not practice-changing right now, but it does mean that we've got to go back and regenerate our hypotheses in renal cell in the first line.